Okay, so we're going to get started. So we're in the research and neuroscience breakout session, the first one in the morning. And just FYI that as Dr. Talebizadeh is speaking, if you have questions, please put them in the Q&A tab and I'll be monitoring those, and we'll ask her those at the end when she finishes. So my name is Olivia Veach and I'm at the University of Kansas Medical Center. And today your speaker is Dr. Zohreh Talebizadeh. She's a professor at Children's Mercy Hospital as well as the University of Missouri, Kansas City. And she is director of the Genetics Research Lab at Children's Mercy. And today she's going to be talking to you about work related to understanding the genetics of autism and thinking outside the box. So good morning, Zohreh.
Hi, Olivia, and hi, everyone. Thanks a lot for the kind introduction, and thanks, Matt and the colleagues at KU for inviting me to this fantastic conference, and I apologize for the technical issues in the beginning. So I'm going to, as Olivia said, I'm going to talk about genetics of autism. My autism research is focused on 3 areas: subtyping, females with autism, and gene regulatory factors. And I divided my presentation into 2 parts. In part 1, I will briefly talk about few examples of genetic research studies. And then in a second part of my presentation, I would like to share with you how I'm trying to use lessons that I have learned from basic research to develop an outcomes research project for autism. So let's start with some background information. What's the difference between genetic and epigenetic alterations? The simple answer is that genetic mutations disrupt the function of a gene, but epigenetic defects misregulate gene expression. Gene expression could be regulated by mechanisms such as DNA methylation, non-coding RNAs, or alternative splicing. There are other processes too. I just listed these because throughout my presentation, I have a few examples related to these mechanisms. Environmental factors may also influence on epigenetic machinery in our body, including factors such as diet, stress, air pollution, smoking, alcohol, or other consumption, or other environmental factors, and it could be prenatal exposure or postnatal exposure. In addition to mutations in DNA coding sequences, abnormalities in gene regulatory processes, including alternative splicing and non-coding RNAs, may also contribute to complex conditions such as autism. Coding sequences of genes are composed of segments called exons, as shown in the figure on the left side of the slide. Alternative splicing is a process by which different assembly of exons may be made, resulting in generating different versions of a gene. And non-coding RNAs, such as microRNAs, do not code for proteins. They are small segments of sequences of the DNA sequences that they bind to the protein coding genes and regulate their expression level. Significant progress has been made in identifying genetic causes of autism as depicted in this figure. But with the exception of a few rare mutations identified as causative genes, majority of the findings are genetic risk factors with unknown function. Of course, there are much more detail than that, and I don't want to, in the interest of time, go through a lot of technical details, but I just wanted to give you a general idea of where we are as far as understanding genetics of autism. The largest genome-wide association study, or GWAS study, for autism identified a genetic variant or a SNP on chromosome 5p or to be associated with autism. This was an important discovery made several years ago, but associated SNP was outside the coding sequences, and its functional role in autism was not clear. The reason I wanted to mention this discovery is that to highlight some of the challenges as far as understanding the function of genetic variants. A few years after that GWAS discovery, a research team led by Dan Campbell used a novel approach by utilizing bioinformatics tools, sequence homology, and gene expression approaches. And they showed that this 5P autism-associated SNP is located inside a non-coding RNA, which has sequence homology to an X-linked protein coding gene. The product produced from this non-coding RNA binds to the protein coding gene on X chromosome, resulting in down-regulation of this X-linked gene. This was a very clever approach, in my opinion, because it demonstrated that how genes located on two different chromosomes, in this case, X chromosome and chromosome 5, may interact with each other and potentially contribute to the etiology of autism. Different approaches have been used to address heterogeneity in autism and to identify subtypes. Many of these studies stratified subjects based on severity along a single domain, such as language impairment or nonverbal communication, while individuals with ASD, as you know, manifest deficiencies across a broad range of behaviors. Valerie Hugh, a good friend of mine and my colleague, took a unique approach to address the heterogeneity and subtyping. She evaluated autism diagnostic interview, or ADIR scores, a broad range of symptoms using multiple clustering methods. Her study resulted in identification of 4 subtypes of autistic individuals. In collaboration with her, I used the Identify ADIR subtyping to reanalyze genotyping data available from about 400 multiplex families. And the multiplex families, again, as you may know, means that they have more than one affected child with autism. To do so, we developed a multi-step stratification method and generated 16 ADIR-related subtypes, as shown in this figure with different colors. And we ran separately linkage analysis for each one of these subtypes. I don't want to go through detail of the subtyping process, but I just wanted to show you that we used multiple steps in order to stratify, and at the end we apply gender. We separated families based on having male only or female containing families at the lower side of this figure. Obviously, subtyping reduced sample sizes. We started with about 400 families at the top, non-stratified one. At the end, as you can see in some of the boxes, we ended up having 16, 8, and 25 much smaller sample sizes. And statistically speaking, we would expect to lose detection power in analysis, but since we actually lowered the existing heterogeneity in the study cohort and generated more homogeneous subtypes, our linkage results actually improved. And we found a subtype-specific linkage, and 2 subtypes showed positive linkage to chromosome 5p14. The same location that I just mentioned in the previous slide about its association with that non-coding RNA. So the next step for this project is that we would like to study the role of the 5p non-coding RNA in these 2 these subtypes that we identified through this stratification method. Now I'm going to switch to another study or another approach. Females have 2 X chromosomes. X chromosome inactivation occurs early in embryonic development in females to achieve gene dosage compensation with males. This is a random process, and usually one of the two X chromosomes in females is randomly inactivated. Deviation from random X chromosome inactivation or skewness may have impact on expression of X chromosome genes in the females. So because of the gender differences in autism, so we find this concept of interest, and we wanted to see what is the X chromosome inactivation patterns in females with autism. So we did a study a few years ago, and we find a higher level of unequal X inactivation or skewness in autistic females, about 29% compared to 11% in control. X inactivation, what I want to highlight is that is a DNA methylation process. And in the next slide, I will show you how we use the X chromosome inactivation pattern as a stratification method in evaluating expression of a gene involved in circadian rhythms. Circadian rhythms are biological blocks regulating physical, mental, and behavioral changes in response to a daily cycle. This system influences body functions such as sleep, awake cycles, eating habits, and digestion. Abnormalities, as you know, in these biological processes have been reported in autism. Key genetic components of circadian rhythms are chromatin remodeling genes that regulate DNA methylation. Recently, using a combination of alternative splicing and sequence homology, we identified a variant for a gene called KDM5C, an X-linked chromatin remodeling gene with a novel variant as shown in the left side of in the right side of this slide, that is originated. This variant we discovered that it is originated from a non-coding RNA, or has a segment originated from non-coding RNA. We showed that this long non-coding RNA is part of a transposed gene with a sequence homology to a parent gene on chromosome 10. So again, I wanted to give you a little bit of picture of when we talk about complexity of a genome, how this type of mechanism could be applied to identify different genes that may have complementary function and may work together. We show that expression level of this novel KDM5C variant is significantly higher in autistic females with an extreme pattern of X chromosome inactivation called, again, XIS, or skewed X chromosome inactivation. This finding is important because chromatin genes are involved in DNA methylation, and as I mentioned, X chromosome inactivation, which we use as a subtyping method, is also a DNA methylation dependent process. So I think this study further demonstrates the importance of using biologically meaningful subtyping in autism genetic studies. Now I'm going to give you one example of gene environment interaction models. As I said earlier, there are some environmental factors that may contribute to autism, and my colleague, Dr. Befferso from MU, has been working on a gene environment model for autism. His team has shown that a combination of exposure to a high level of stress during pregnancy and being a carrier of a stress-related genetic risk factor may increase the risk of having a child with autism. This gene is called SERS, which is a serotonin transporter. And in collaboration with his team, we have shown that stress-related microRNAs may also be differentially expressed in blood samples collected from mothers exposed to high level of stress during pregnancy. This collaborative project highlights the importance of investigating epigenetic factors such as microRNAs to better understand gene-environment interactions in autism. And in this table, I just wanted to highlight the pathways that majority of the predicted genes for those differentially expressed microRNAs may be associated with, and some of the pathways were related to top pathways, dopaminergic and serotonergic pathways, which was of interest because the gene search also is a part of those pathways. So to summarize the first part of my talk, I would like to highlight three points. ASD, as we just discussed, has a complex etiology that includes both genetic and environmental risk factors because of very diverse clinical presentations. Subtyping may be needed to improve study results. Obviously, these are real challenges in studying autism, but they also provide opportunities to create novel approaches and establish multidisciplinary collaborations. In the next or second part of my presentation, I would like to discuss the importance of multidisciplinary approaches. Despite significant progress made in understanding the etiology of autism, there are no effective biomarkers to facilitate the early detection and effective treatment. Two strategies have been commonly used to address these barriers. One has been to increase sample size in research studies to improve statistical power, and the second approach has been to developing a new analytical tools to improve, again, methodological aspects. And these strategies have been helpful in some cases, but translation of research from bench to bedside is still a major challenge, as you know. So I am, again, as you know, I am a basic researcher, and the focus of my research project is on identifying genetic autism-related genetic and epigenetic risk factors. But a few years ago, almost accidentally, I saw a request for proposal from a funding agency called PCORI. I learned that they were not interested on funding basic research, but something captured my attention. They highlighted the need to change the culture in research and making it more patient-driven by engaging patients and other stakeholders in research study design. This was a relatively kind of new topic for me, and I thought that this may help me to find the missing piece that I was looking for. So I began educating actually myself about outcomes research because I was not familiar with that discipline. And in 2016 and 17, I applied for and received 2 awards from PCORI to start doing some engagement work for autism. First study was an engagement project. What I proposed was to start a dialogue about building a bridge between genetics and outcomes research. We formed a community advisory board, including parents, advocates, and use a variety of participatory methods, including focus groups, surveys, to identify gaps, barriers, and facilitators. We identified that one key barrier is the lack of interactions or communication between researchers from these 2 disciplines. So our goals for this study, which we call it Autism Genetics Outcomes, or OUTGO, was to create a hybrid concept connecting the outcomes and genetics disciplines to demonstrate the potential and benefits of interplay between these 2 disciplines. And we thought that by doing so, it may stimulate much needed dialogue between different stakeholders, and it may facilitate finding practical ways to incorporate patient and parents' perspectives when designing genetics outcomes or GO studies. And we refer to GO projects as projects integrated research approaches that utilize data and principles of both genetics and outcomes research. And in the next step, we began creating a conceptual framework for developing GO research hypothesis. How do we develop GO hypothesis? We developed a semi-structured literature review process to collect and process feedback from our study participants to evaluate research findings in the literatures from different perspectives and identify potential geo-hypotheses that may arise from these findings as shown in this workflow. I don't want to go through the detail. I just wanted to give you a general idea that it involved multiple steps and also several evaluation phases. And we formed advisory boards, including 30 members, and as I said, including clinicians and patient advocates. This was the first efforts to demonstrate that it is doable to connect stakeholders' perspectives with research findings. This project was extremely time-consuming because there was no model to rely on, and we had to build the conceptual model from scratch. And obviously, I don't claim that this is the best model, but this is a beginning of, I think, hopefully moving forward to further refining this model. And on the right side, I just wanted to show you that we practice this model, this engagement model with our stakeholders, and we reviewed about 60 papers, and based on their comments, we identify about 10 potential geo projects or hypotheses for autism, and then at the end, we developed one geo hypothesis for depression in autism with the feedback that we received from our stakeholders. So if I want to summarize what I have learned from this long journey of becoming a kind of a double major is one thing. I realized that both research community and patient advocates share the same concern over the need to make research more patient-centered. But this concept is described or defined differently from different for different target audience. As you know, there are different terminologies being used, including precision medicine, translational research, community-based research, team science. Obviously, each terminology has a different meaning, but they all need to utilize to some degree both basic research and outcomes research, but there are no actionable protocols or roadmap to show how to build the bridge between these two essential disciplines. Typically, genetics research studies rely on quantitative methods. On the other hand, in outcomes research, the most commonly used methods are qualitative. Despite differences between the two research approaches in terms of hypothesis generation, collecting evidence, and or overall objectives, their common purpose is to make research more translational, either through improving health outcomes or contributing to precision medicine. In the outgo initiatives that we developed, we attempted to show that there is a need to incorporate patient and parents' perspectives in study design, and using mixed methods may facilitate moving the autism genetics field closer to the center, which I mean by center, more closer to translational research. So, but why do we need to incorporate patient perspectives? Let me briefly describe two examples to describe this question. Parents have frequently raised concerns about GI issues in autism. Ellen Balti, a mother of a boy with autism, first brought the attention to a potential connection between her child's rapid developmental and behavioral regression and his GI symptoms. She had no formal medical or scientific background, but started to study medical literature in an attempt to find a solution that would ease her child's condition. In 1998, she published a perspective paper. This and similar concerns raised by other parents contributed to bringing awareness in the research community about the potential co-occurrence of GI issues in autism. And in 2009, association between a gene and a gene called MET in families with co-occurring autism and GI conditions was discovered. This gene has a dual function. It contributes to both GI functions and brain development. Subsequently, more attention has been given to the importance of targeting GI problems in autism, particularly for nonverbal cases. In the second example that I want to briefly mention is that for many years, the parents of children with autism have reported that behavioral symptoms diminished when the child had fever. A study published in 2007 reported that some children with ASD may show behavioral improvements during fever. Subsequently, functional and mice studies resulted in discovering the cellular mechanisms of this so-called fever effect. Later, the results of a smaller clinical trial showed that a nutritional compound may improve symptoms of autism, at least in some cases, by mimicking what happens in the body during a fever. What I wanted to highlight by going quickly over these two examples is that both of these two examples, in my opinion, have two common elements. One is that in both examples, the discoveries resulted in improving, or at least the possibility of improving patient health outcomes. And the awareness about both of these issues, GI issues and fever effect, initially was noted by parents. So obviously not every observation or every anecdotal observation has a biological or mechanistic basis, but some do. So my interest as a researcher is that is there any way how to in a more systematic way capture and synthesize those type of observations? I don't know what's the answer, but I hope we could find a way. So based on my experience working with both fields, one of the major barriers, I think, is that collaboration has a different meaning and nature in basic research and outcomes research. As I summarize here, again, I think that there are different cultures and different basically approaches, principles involved in both of these line of principles, and in order to make this type of collaboration happen, we need to come up with ways to better make a bridge between these two line of research. Taken together, I think there is a great need to define the interactions between basic research and outcomes research in order to further promote patient-centered research in autism. And this integrated concept, I don't think it exists actually, even though we keep talking about that, and it needs to be developed. So we could, I think, take the lead in this initiative by leveraging the existing but disconnected strong parent advocacy efforts and unique clinical and research and technology resources in our region. And I listed here a few of those that I'm sure you are familiar with many more resources that we have. And of course, this type of projects requires resources and funding. I applied for a PCORI funding to develop strategic planning for this, for developing a kind of a strategic planning for this, for a platform, or that I call it consortium, to start building, basically figuring out how can we achieve this type of integrated approaches. And I think actually I wanted to say that just a few days ago I learned that my PCORI grant was funded. Contract is still pending, but the project will start in June. And here I wanted to list some of the anticipated outcomes from this capacity building project. In addition to linking disconnected efforts and resources, I think that obviously we working on this type of integrated works would help us to reduce duplicated efforts and promote synergistic, impactful research programs. And it may also lead to developing new educational opportunities and obviously developing a novel integrated approach concept that I don't think it does exist. So it is exciting that the project was funded, and I look forward to start working on that with many of you actually in this meeting who are part of that. In summary, I wanted to kind of summarize what I covered quickly in my presentation in this slide is that autism, as we all know, is a complex heterogeneous condition. As a result, there is a great need for thinking outside the box approaches. And we need to evaluate data through different lens and from different perspectives. I just quickly went over a few examples to show you what I mean by not thinking outside the box approaches and integrated methods. This, of course, requires also investing on developing novel integrated analytical concepts. So in my opinion, there is no shortage of tools, software, and machines. Of course, it would be nice to develop even more advanced tools that we currently have available. But what I think the field of autism and perhaps other complex conditions is in great need is the shortage of dedicated time to kind of manually inspect and connect different pieces of data and knowledge and not just rely on tools and machines, because there are so many details that we may not be able to capture just without including the human factor. And we usually talk about state-of-the-art technologies as our resources. And I hope that someday we could also highlight access to a state-of-the-art conceptual framework and how they may promote patient-centered autism research, as I, again, for the next second part of my presentation, I try to demonstrate to you what I mean by patient-centered autism research. And I hope that this autism research consortium that we plan to establish, I hope it could contribute to reaching this goal. Of course, this is, as I said, a very tall goal, but I am pleased and excited to see that we receive funding to at least start developing the strategic planning and working together to, again, see how we could achieve this much needed, in my opinion, goal. So this, and here I wanted to acknowledge my collaborators and also funding agencies for supporting our research projects. And I'm gonna stop here, and I hope we should have time, hopefully, for question. If anybody has question, happy to take your questions.
Great.
So should I stop sharing, I believe?
Yeah, that sounds good. And then maybe you can turn your video back on so we can see your lovely face.
Oh my gosh, I don't know about that, but let me see if I can figure it out, my dear. Okay, don't give me, okay. How about that?
Wonderful.
Hey,
Yay, great.
But mission accomplished.
Yeah, that was a great talk. Very inspiring. And I have a couple of questions both from Dr. Mosconi. So I'm going to start with his first question, which is more related to the end of your talk, and then we'll go to his second question, which is more toward the beginning. So In your experience with the OTGO project, were there consistent areas in which patient advocates or other community perspectives altered approaches for developing or translating science?
This is a great question. I have to admit that we just started working with patient and other stakeholders, right? So I don't want to claim that we exhausted basically the resources. So the short answer is that yes, we there were some scenarios, some situations that actually I'm trying to see if I can find one example. I may remember that later on. I will tell you, Matt. But there were scenarios that not exactly we changed the direction, but for example, like those two research studies that I mentioned about GI and fever, as you know. There were observations like that, that when I heard the concern or the question in mind of parents, for me as a researcher, it helped me to think about, oh, maybe I need to also look into this potential connection. Because if I didn't have that opportunity to hear that, and if I just rely on what I found in literature, I'm not saying that, I mean, we don't have enough papers in the, scientific literature. No, there are plenty of them, but if something has not been studied, then obviously it's not being published. And if it is not there, if my only source of data is literature, then I won't have any way to become aware of that. So the short answer, hopefully, is that yes, I realize that there are some cases and scenarios that we may hear some get some feedback that would help us further refine our approaches.
So before I move on to the next question that Matt posed, I was wondering, so I just happen to be privy, since I collaborate with you, to a project you're doing focusing on BMI and understanding potential effects of prescription associated weight gain. And I was curious if that project idea stemmed from parents or clinicians that you'd spoken with as you were working on this OTGO project, and how that kind of came to, yeah.
Great question, Olivia. That study, that actually I didn't mention anything about that because of the interest of time, but a few years ago, actually, I had a conversation with one of our clinicians, and she actually brought up that topic to my attention, that in her clinic, she observed that so many of her patients with on the spectrum also, either themselves or someone in their family, also happen to have metabolic related conditions or obesity. And then when we look at the literature, I didn't find a whole lot of information in relation to the prevalence of obesity back then a few years ago, but I found a few papers that look into the prevalence of obesity and overweight in this population, and there were some suggestion that there might be a higher prevalence of obesity. So that actually captured my attention, and that led that conversation again that was started by observation from one of my clinicians, and then connecting that to couple papers and findings in those papers. Then actually we developed a research questions to look into some of the genetic risk factors associated with BMI and with obesity in the autism. So, and we find actually some interesting finding that we think that it suggests that at least in a subset of individuals with both autism and obesity, they may have some underlying metabolic conditions. Again, not for all of them, but some small subset. And to me, again, that is encouraging because that may help better developing more targeted treatments, at least for those specific subtype.
Okay, so that actually leads perfectly into Matt's next question. So he asks if you could clarify whether the ADI measurement subtypes that were identified through the clustering from Dr. Hugh have been replicated in independent data sets.
You asked a lot of good questions, Matt. I wish Valerie was here, but I am not sure, to be honest with you. I do know one thing, that Valerie herself, she was able to replicate that into another cohort, completely independent cohort, because for that original study that she published, and I referenced that, she used data from AGRI. As you know, AGRI has multiplex families. And then later on, I don't know if she had a chance to publish it yet, but she kind of replicate that in data in Simon's simplex families. And she kind of saw similar kind of direction as far as different subtypes. And again, I don't claim, and of course I shouldn't say that because that part of the work was her, of course, research, but I don't think she also claimed that this is the best or the only stratification method. But what we wanted to show, and what I wanted to show by bringing that piece into my presentation, was that doesn't matter, in my opinion, even as long as it's meaningful, of course, what kind of stratification methods we use. If we use some type of stratification methods that has, of course, some scientific or biological or some type of basis, of course, and merit into that, even though we ended up losing obviously sample size, but it would increase the likelihood of finding hopefully something more meaningful in relation to small subtypes. Again, that is one approach, and I'm sure there are plenty other approaches. Yeah, but what I like specifically about that ADR approach that she took was that, as you know, instead of focusing on one domain, she didn't basically she didn't pick some specific domains. She just applied uniformly clustering analysis on all the data that she had, and I think that gave her maybe a little bit less biased kind of approach. So that's one way.
Yeah, no, I think it's an important and powerful approach. I mean, the reason I bring up the question is because, this is just, we're all sort of looking for these ways to start parsing clinical and outcome heterogeneity so that we can better understand etiological pathways and mechanisms, because just looking at, binary distinction of autism or not just doesn't, we know that that's not a very robust or replicable, reproducible way of approaching these. So we're just all looking for clustering methods or ways of identifying, robust dimensional traits in ways that can be reproduced and therefore really meaningful and robust. So that's why I'm asking. I'm hopeful that we can reproduce those results. And I think of other, methods as well. I think of Olivia's work, and she's really focused on sleep, and that is just an important and it's an important and potentially powerful target for phenotyping, identifying distinct pathways, and there just are so many targets that could be enveloped into these subgrouping methods, and so we have something that's being reproduced. That's awesome. That'd be great.
Exactly. Exactly. It is, again, it's not without limitations, of course, as you know, and reproducibility, as you mentioned, is going to be one challenge for sure, because when you kind of, again, I can't say customize, but it requires, right, applying some kind of a more fine-tuning elements that it makes it harder to exactly be able to replicate the same kind of study. But on the other hand, I don't know, I think in the interest of working more efficiently also, that's also is the approach. And plus, again, as you said, what we know about autism, whether we want to take the approach, address heterogeneity or not, it is a highly heterogeneous condition. And it's not just the clinical side, as I try to quickly mention in my presentation. Genetic part of it is also complex too. It's not just phenotypic presentation.
Okay, so I didn't see any more questions in the chat, and we're at 10:54, so we came right up on a time. And so yes, thank you everybody for attending. Again, great talk, Zohreh, and we hope that everybody has a good day. I'll be seeing some of you all later. So yeah.
Thank you. Thank you, everyone. See you later.
Bye.